Okay, so in this video, we're going to talk about control groups when you're setting up a controlled experiment. So a control group is oftentimes used by researchers or scientists in an experiment as a comparison group. So it does not receive treatment or it's not changed. Like when you're changing the independent variable in your experiment, it's not applied to this control group because this control group is used as a comparison for how the other groups do. So let's look at some examples. So let's say in another experiment, you are curious about whether or not heat increases the speed of a chemical reaction. So you set one flask on a hot plate and the other is not given any heat. And then you observe and record the results. So when you look at this experiment here, our control group is the group that is not having a variable be changed. So like in this experiment, the independent variable is the change in temperature. So we're applying heat. That's what we're curious about. So here, this control group doesn't have any heat applied, so we can compare, well, how does the heat change it, right? So it's a comparison group in this example. So you add, and then we have our experimental group. So you add whatever chemical you want to see react, you start your timer, and then let the reaction go. And in this animation, you can see how the experimental group, how the heat actually did increase the speed of the reaction. So the control group in an experiment is separated from the rest and does not have any changes in the variables. The independent variable changes in the experimental groups, but not in the control group. And this shows like if the independent variable actually has an impact or an effect on your experiment. The control group is allowing you to compare. Okay, so let's pretend one day you notice that when you cut an apple slice, it turns brown pretty quickly once it's exposed to air. And you're curious if adding lemon juice will stop them from turning brown. So you design an experiment that will test the effectiveness of lemon juice at different concentrations. So you get some solutions that are 25%, 50, 75, and 100% lemon juice. And you get some apple slices, but you also are going to set up a control group. In this example here, the lemon juice, the different concentrations of lemon juice, that is our independent variable. And we're curious, our dependent variable is how brown the apples turn. Like that's what we will be measuring. So the control group, though, is to ensure that our experiment is actually working, like as a baseline for comparison. Now, the reason you can't use, like if I circle this right here, so the reason that we're not just using this apple as our control group is because in these, we are actually adding a liquid to the apples. So in experimental design, it's super important to have not a bunch, but to have controlled variables. So if I add a liquid to these 4 apple slices, then I need to add a liquid to this one. That's part of my controlled experiment. But the difference between my control group and my experimental groups is what is that liquid, right? So I'm testing the different concentrations of lemon juice, but I'm going to compare it to one without lemon juice, which is my control group here. So let me go ahead and just erase all this. So let's go ahead and see then. So you do the experiment and you notice that there is actually some impact. The lemon juice does have it does have an impact, on preventing the apples from turning brown. Okay, so you do this experiment, you're like, oh, 100% lemon juice is the most effective. Look at my apples didn't turn brown. But it's also important when doing experiments that you repeat your experiments over and over and over. You have multiple trials to confirm your data and to confirm your results. Okay, so that's something that students often forget. It's important to repeat your experiment and have repeatability and multiple trials to draw your conclusions. Okay, so let's say this is my last example on control groups, that you're curious about the effect of different nutrients on plant growth. So you want to test whether a nitrogen-based fertilizer or a phosphorus-based fertilizer is better for plant growth. So you start with 3 pots of even size. And in one pot, you just take soil from your backyard and you don't add any fertilizer. In the next pot, you take soil from your backyard and you add some phosphorus-based fertilizer. And in the third pot, you take the backyard soil, dirt from your backyard, and you add a nitrogen-based fertilizer. So when I look here at these pots, I can figure out that my independent variable, the thing that I am changing or curious about, is the type of fertilizer. All right, so then I plant seeds in each of the pots of the same species. I apply the same amount of water to each of the pots, another controlled variable, and then I allow them to grow in the sunshine, right? So there's my no fertilizer plant, my phosphorus-based fertilizer, and my nitrogen-based fertilizer. So then here you could record your data and you could draw your conclusions. Now from looking at this, you would still have to apply like statistics and data analysis, but you can see how adding fertilizer did increase plant growth. And then you can draw conclusions from that saying, well, the nitrogen-based fertilizer had a better impact or greater impact on plant growth, right? But you have your no fertilizer pot as your control group. That is for comparison. So here you can say that fertilizers do increase plant growth in your experimental groups. But now of those fertilizers, we can see that the nitrogen-based fertilizer was the best at increasing plant growth. But again, this is just 3 pots. So if you want to have valid conclusions, it is super important to repeat your experiment multiple times, have multiple trials, have large sample size, so you can draw valid conclusions from your results. Okay, that's my conversation on control groups. Control groups are used in experiments for a comparison. They are not altered by the independent variables in the experiment, but they do share the same controlled variables. So in this experiment, they had the same seed, the same water, the same size pot, the same sunshine, but they did not get the treatments. They're just used for comparison. Okay, all right, good job.